Hello everyone again. Today we're going to do meiosis part 2. In meiosis part 1, that presentation, we talked about the process of meiosis, which is the process used for making gametes. And gametes are sperm and egg cells. So it's used in only making sperm and egg cells. And at the end of meiosis, you have a haploid cell with 23 chromosomes. So there would be 23 chromosomes in the sperm, 23 in the egg. They come together to form the fertilized zygote, which will now have 46 chromosomes. What we're going to talk about today is when meiosis doesn't occur properly and you get sperm and egg cells resulting that do not have the correct number of chromosomes. So when you do have the correct number of chromosomes, that's called euploidy. So for humans, a euploid cell, a sperm or egg cell, would have 23 chromosomes. There are conditions in which there's an abnormal number of chromosomes, and that's called aneuploidy. So that would be an abnormal number of chromosomes. So normally you have 23 in the sperm, 23 in the egg, and they come together to form 46. In aneuploidy, you have a fertilized egg or a zygote that does not have 46. If they have one less chromosome, that would mean they would have 45. And that's called a monosomy. So a monosomy is when for one of the chromosomes, one is missing. And as a result of one missing chromosome, the total number of chromosomes is now only 45. The opposite kind of problem is called a trisomy. And trisomy is when for one chromosome you have an extra. So you would have one chromosome that has 3 instead of 2, resulting in a total of 47 chromosomes, and that's called a trisomy. So both a monosomy and a trisomy are forms of aneuploidy, an abnormal number of chromosomes. Aneuploidy results from errors occurring during meiosis, and it's called non-disjunction. And that's kind of a hard word to understand. Normally in meiosis, your sister chromatids pull apart. If they don't pull apart, that means one cell is going to get 2 and another cell is going to get none. So let me show you in the next slide. So this is examples of non-disjunction. So it can happen either in meiosis 1 or meiosis 2. So this first one is showing meiosis 2 here. And here we have meiosis 1 occurs. We have 2 daughter cells. They're just showing one chromosome that has 2 sister chromatids for the chromosome. Okay, so that's here. And then in meiosis 2, those sister chromatids should split and one go to one daughter cell and one go to the other daughter cell. Then when that egg is fertilized by a sperm, you have 2 of that chromosome, which is the normal number. In non-disjunction, those 2 sister chromatids don't split. So one daughter cell gets both of those. The other daughter cell gets none of those. And remember, this is just one of your 23 chromosome pairs. So when that cell is fertilized by a sperm, you get 3 of that chromosome because you have 2 from the egg and 1 from the sperm. So that's a trisomy. And when this egg, if it should get fertilized by a sperm, you only have 1 of that chromosome because the egg didn't have any, and that would be a monosomy. It can also happen in meiosis 2, or excuse me, in meiosis 1, where you would have one cell that didn't get a copy of a chromosome, and again, at the result, you would have some fertilized cells that have trisomies and some that have a monosomy. The way that these chromosome disorders are diagnosed is with something called a karyotype. And a karyotype is when from an individual, like you could have a karyotype done of your cells. They would take samples of cells. In humans, they use white blood cells usually. The reason they don't use red blood cells, excuse me, is that red blood cells don't have nuclei, so they don't have chromosomes. And since chromosomes are only visible in the X form during mitosis, they actually treat them with a compound that causes the cells to enter into mitosis. And then from there, they can do a procedure where they kind of freeze the cells in mitosis and then isolate the chromosomes and get a picture of the individual's chromosomes. If this is done, this is not done as commonly in adults as in a fetal blood sample or a cell sample, because non-disjunction is not a genetically inherited condition. So it would happen in a particular egg or sperm that resulted in that embryo forming. So if you were going to diagnose a pregnancy, whether there was a chromosomal abnormality, you would need fetal cells. Maternal or paternal cells would not tell you whether or not there was an abnormality in the fetus. And so in order to get fetal cells, the most common method is an amniocentesis. And in an amniocentesis, they use a needle to take a sample of amniotic fluid, and that fluid has fetal cells that have kind of sloughed off like your skin cells do and are suspended in the fluid, so they can extract those cells and then do a karyotype. This here is, they use an ultrasound to locate the position of the fetus so that when they put the needle in, they don't hit the fetus, that they know they're just hitting the sac. And so from those, they can produce a karyotype. And this is a karyotype here. And so what it is a picture of an individual's chromosomes. And the chromosomes are all known as in terms of size and what genes they have. And so this picture represents a particular individual's 23 pairs of chromosomes. Remember, you have 23 pairs, 46 chromosomes altogether. So each of these represents a pair. And so there's 22 pairs that are called autosomes. And autosomes, I'll write that word down, are everything but are the chromosomes that everybody has the same. The final pair, the 23rd pair, are the sex chromosomes, and they're the ones, the two chromosomes that determine the sex of the fetus. And the two options are two X chromosomes, which is a female, we'll talk more about this later, or an X and a Y, which would be a male. So this individual has two X chromosomes, so this is a karyotype of a female. The way the chromosomes are arranged in the karyotype are by certain features, and they're arranged by the length. So if you go back, when I do this narrations, it doesn't permit me to go backwards, so I can't go back and show you that again. But they're arranged by size. So chromosome number 1 is the biggest chromosome. Chromosome 22 is the smallest chromosome. And then they're stained, and then they have a particular pattern of bands. So that's another way that they're arranged or matched when you do a karyotype. And then the arms. The arms are the pieces above and below the centromere. So the centromere is the position in the middle. And so that varies in chromosomes. So sometimes it's close to the top, sometimes it's in the middle. And then the arms above the centromere are called P arms, and the ones below the centromere are Q arms. So those are the three criteria that are used for matching the chromosomes. So as I mentioned, we're going to talk now about known aneuploidies that occur in humans. And they can occur either in the sex chromosomes or in the autosomes. So the autosomes, again, are chromosomes 1 to 22. So males and females have the same autosomes because it's only the sex chromosomes that are different between males and females. So for the autosomes, which represent chromosomes 1 to 22, there are no known monosomies. So no monosomies that might form during a fertilization. So that's with a fetus that would be missing a chromosome, survive. So they would result in miscarriages. And there are a few autosomal trisomies that can result in a live birth, but not that many. In most cases, an autosomal aneuploidy, to use the right terminology, which is an abnormal number of autosomal chromosomes, does not result in a live birth. And so most cases where this happens, it's not a survivable fertilization. And there are just a few cases that have been known to survive, although they're rare and the survival rate is low. And the three that we'll talk about are all trisomies because, again, there are no autosomal monosomies. So there's no one born that's missing one of chromosome 1 to 22. And you might think, well, why is a trisomy bad? Because you have an extra chromosome. So you're not missing anything, but instead of having 2 of that chromosome, you actually have 3. But our body's really geared, and remember, genes are the instructions for making proteins. And the way our cell is designed to work is to have 2 copies of every gene. And having either 1 copy, which would be a monosomy, or 3 copies, which would be a trisomy, results in abnormalities. So even though you have an extra copy of genes, that can lead to excessive productions of certain proteins that can lead to problems for that fetus. So the 3 trisomies, and you probably have heard at least one of these that do exist in humans, are trisomy 13, which is also called Patau's syndrome. This one's very rare. Trisomy 18, which is called Edwards syndrome, and trisomy 21, which is Down syndrome. So I'm going to just talk briefly about each of those. Excuse me. Trisomy 21, which is Down syndrome. So these individuals are born with 3 copies of the 21st chromosome. And that leads to a number of characteristic abnormalities, some of which are shown here: short stature, some changes in the face, eyelids fold, a flat face, usually significant intellectual abnormalities, stubby fingers, a gap between the first and second toes. So individuals born with Down syndrome tend to look alike because they have the same abnormal facial features. And this is the most common of the trisomies, and it's a trisomy 21. So this would be a karyotype of an individual with Down syndrome where they have 3 copies of chromosome 21 and everything else is normal. Patau syndrome results in more severe abnormalities. It's trisomy of 13. So individuals with Patau syndrome have 3 copies of the 13th chromosome. Also the severe intellectual as well as developmental abnormalities. And then the third one is Edwards syndrome, and Edwards is trisomy 18. So they have 3 copies of the 18th chromosome. Again, characteristic facial abnormalities as well as major problems in a number of organ developments, and have a significantly shortened lifespan. So those are the 3 aneuploidies associated with autosomal chromosomes. There are 2 others associated with the sex chromosomes, again, which are the XY for a male, XX for a female. And it's the same thing. If you're missing one of them, that would be a monosomy. If you have an extra one, it's a trisomy. And in this case, there is the only known monosomy in humans that is survivable is in with the sex chromosome. It's called Turner syndrome. And Turner syndromes are females with only 1 X chromosome. So there's only 1. They don't have the second. That's why the O is here. It means none. They don't have, there's no O chromosome. And so they have 1 X chromosome. And we know humans can survive with 1 X chromosome because males only have 1 X chromosome. And they have some changes in features and some impaired reproductive abilities, but can lead a normal lifespan. Klinefelter syndrome is in males, and it's males with an extra X chromosome. So instead of being XY, they are XXY, so it's a trisomy. And men with Klinefelter syndrome tend to have poorly developed male sexual organs, more female-like characteristics. So they sometimes have sterility problems, have some breast development, kind of body hair patterns more associated with females. It might not be so severe that it's noticeable. And so sometimes men with Klinefelter syndrome, it's not really diagnosed until they have problems in reproducing because of sterility, and then they find out that they have Klinefelter syndrome. So this would be a karyotype of a Turner syndrome. There's only 1 X chromosome and there's no second sex chromosome here. It's just the 1 X. And this would be Klinefelter's, where there's 2 X's and 1 Y. In the aneuploidies that we talked about, results in either an extra chromosome like Down syndrome, Klinefelter's, or missing a chromosome. So it's a whole chromosome that's either an extra one or you're missing one. There are a few abnormalities where you're not really missing a chromosome or have an extra, but pieces of chromosome are either missing or copied. And that usually happens during crossing over. Remember, crossing over is when the chromosomes switch pieces. So if one gives a piece to another chromosome, but that one doesn't give a piece back, then you can have a chromosomal abnormalities. Again, these are not inherited. These slides discuss the types, but I'm going to go to the pictures to discuss because it's easier. So one type is a deletion. And a deletion is where a chromosome is missing a piece. So see, this is kind of the letters just are to indicate location. So this chromosome here is missing that top piece. That's called a deletion. And so whatever genes are in that piece of the chromosome, that chromosome doesn't have those genes. This one is a duplication. So you can see there should just be this section is here twice. So those genes are their extra copies, and again, that can be problematic. This one is an inversion where the piece of the chromosome is upside down. So that means genes aren't where they're supposed to be. And so that can lead to abnormal function of those genes. And then the fourth one is a translocation. And a translocation is when crossing over occurs between non-homologous chromosomes. So usually crossing over or between chromosomes that have the same genes, but here we have ones that don't. And so you get genes on a chromosome where they don't belong. So this one has QR instead of GH, and that's called translocation. There are examples of all of these causing various syndromes in humans. Sometimes they can be not particularly problematic, but it kind of depends on the size of the abnormality and what genes are affected. I'm just going to show you a couple of them. Williams syndrome is one that occurs in humans, and it's a deletion of a piece of chromosome 7. And this deletion results in some facial abnormalities, some learning disabilities, some cardiovascular problems. One of the interesting things with Williams syndrome, this little girl here is the one with Williams syndrome. And so you can see her facial abnormalities. They are also very happy, engaging, outgoing children. They are very friendly. They have good artistic ability, laugh a lot, are very happy. So that's one of the characteristics associated with Williams syndrome. This one is also a deletion. It's a deletion of a piece of chromosome number 5. It's called cri-du-chat, which means in French cat cry syndrome. And that was named after a doctor that, excuse me, thought that infants born with this syndrome had a distinct sound when they cried that he thought sounded like a cat. And so he named it cat cry syndrome. And that name is kind of stuck with it. This also is associated with severe abnormalities and intellectual impairment, as well as organ and facial abnormalities. And then the last one is a translocation, where a piece of chromosome 8 gets switched with chromosome 14. And this one actually is associated with a type of cancer called Burkitt's lymphoma. It's a cancer of the lymphatic system, the lymph glands, usually in the face. So this little boy here has a tumor here growing in his lymph node on that face side of his face. And that's one of those regulatory genes that get mixed up with the movement of the piece of the chromosome isn't there to control it anymore. So that's the end of the abnormalities associated. One thing that I forgot to mention when I was talking about them is that although in theory the aneuploidies can occur in either a sperm or egg cell during meiosis, they actually most commonly are associated with the egg, so that of abnormalities occurring in the egg. And the older the maternal age of a woman when she gets pregnant, the risk of some of those chromosomal abnormalities increases markedly. So it's often recommended for women that get pregnant over the age of 35 that they have an amniocentesis done to look at whether or not the fetus has any of the chromosomal abnormalities. At that point, it cannot be corrected. It would just being aware that it existed. That's the end of this presentation on meiosis. After you view this, you can take the meiosis part 2 quiz, and we're going to move on after this to genetics. Bye.